Hi guys, welcome to section C for chapter 1. There's a lot of notes in this video, so definitely feel free to pause it to give you a chance to copy what you miss. A lot of notes though. So get your paper ready, turn to section C in the chapter, and let's go. All right, so kind of some background on life science. We're going to go through a lot of scientific terms today. One such term is data. That's just what we collect. It's information that we collect when we're conducting experiments or making observations. There's two different types of data that come into play. One is qualitative, which is a descriptive type of data. It describes the quality of something. If you're looking at a plant and you describe the color, the shape, anything that uses words to describe it would be qualitative data. And then we get into quantitative data, which is really a more common one. That's numbers, that's measurements. It's talking about the quantity of something, the size of the leaf, the number of leaves, the height of the plant. That would all be an example of quantitative data. Then we get into some other terms. Theory and law are two that come up a lot. Law always sounds a little more set in stone, and a theory is more of this just kind of idea. But they're both pretty well tested and pretty well explained in science. A theory explains the why something occurs, and a law explains what's occurring. Okay. Now, in biology, there's not a lot of laws. There's a lot of theories, but there's not too many laws. I'm not sure why that is, but a theory, an example of a theory would be the theory of evolution. It explains why all living things exist. It's not proven. There's data that supports it, but it's not completely proven. Same thing with creation. We believe that we have data that supports creation as well, but it's also not proven. They're both just theories that explain the why. Neither one of those are laws, though. We use all this stuff. We use all this data and these theories and these laws and our observations and whatnot to create a model, which is just kind of this overview explanation of what's being observed in the world around us. We definitely want to keep our worldview, our lenses in mind when we make a model. The secular model would be involved with like evolution, and that's a big picture explanation of the existence of living things. The Christian model would involve creation with that, okay? So just a review on the scientific method. You guys should be familiar with this. The number one step is to form a question about what you see happening around you. What are you curious about? What do you want to know why it is that it's happening, okay? After you come up with that question, you want to do some research to kind of find out what's known about it. And if there's not much known about it, if things haven't already been proven or discovered about it, you want to form a hypothesis. You want to take a guess as to why it is what's happening is happening, okay? But then you need to kind of come up with an experiment to test your idea, okay? This experiment should be considered a controlled experiment, which I'll talk to you more about on the next slide with some additional definitions there. Basically, in general, a controlled experiment is testing only one variable at a time. If you're testing too many variables and you have a difference in your experiment, you don't know what to attribute that difference to. So only one thing at a time. Then you collect your data, both your descriptive, your quantitative, and your measured, your qualitative data. And then you use that data to make a conclusion. You're going to analyze it, you're going to study it, you're going to see how it fits with your hypothesis, and then you're going to come back and make a conclusion that circles back to your hypothesis. Was it right or was it wrong? If it's wrong, it doesn't mean you failed. A disproven hypothesis is every bit as valuable as a proven hypothesis. And then finally, you want to share it. You want to publish it. You want other scientists to be able to see it, follow it, repeat it, and learn from it as well. Okay? So here's this controlled experiment. Really, I suggest that you pause the video at this point and copy these down, okay? But I'll explain it kind of briefly here. A variable is the factor that changes between tests. So let's say I'm going to test the effects of Miracle-Gro on a plant. So I'm going to have two plants. I'm going to have one plant that's my control group that I'm not changing anything on. Then I'm going to have a second plant that I'm going to give Miracle-Gro to. So the only difference between those two is that variable, okay? The single thing that I've changed between the two is more specifically known as an independent variable, okay? Everything else is going to be the same. The type of plant, the type of soil, the amount of sun it gets, the amount of water it gets, the amount of food it gets, everything else has to be the same. Otherwise, if I see differences in the two plants, I don't know what to attribute that difference to. I have to be able to attribute it to the Miracle-Gro, the variable that I changed, okay? Now, if I do see a difference, let's assume that the Miracle-Gro one grows more. That would be the dependent variable. That's something that changes as a result of my independent variable, okay? The control group, again, is the plant that had no difference to it, had no variable. It was just kind of set up to have something to compare to. And the experimental group is the one that has the variable to it, okay? So that's how you set up a controlled experiment. It's really important. Okay, let's talk about life more. There is an abundance of life on the planet, and so we have to classify it. We have to be able to put it into groups, okay? And you can see this list of, 8 different categories of life, okay? And everything that's in a similar category has similar characteristics, okay? As you go down, you can kind of see this funnel shape. As you go down that flow, less things will fit into it because less things have similar characteristics. For instance, if I start at the domain, that's just about all living organisms will fit into that. But as I funnel my way down, I'm getting more specific to all the way to the end of which is the species, which is just a single type of organism. There's one type of human, but there's any number type of animals. Okay, I'll show you another example of that as we go. Let's keep talking about these classifications. Taxonomy is basically the study of classification. It's a type of science that works on sorting these organisms into these groups. Okay. Now, this nonsense sentence, "Dear King Philip came over from Germany safely," is to help you remember. Let me get my pen here. It's to help you remember these. Where's my pen? There we go. I got it. Okay. It's to help you remember these names. So if you notice, the D for deer is domain, king is kingdom, Philip is phylum, came is class, over is order, from is family, Germany is genus, and safely is species. This is a mnemonic. It's just a device that helps you memorize things. It's harder to memorize the order of all those new words. If you can memorize that sentence, it helps you at least put them into order, okay? So we've got domain, which is the broadest. Kingdom is still pretty broad, and then we get more specific. There are different characteristics. There's 3 different characteristics that scientists look at in order to put them into a domain or a kingdom. The first is the type of cell they have. Some cells have nucleus, which is the center part of the cell where the DNA is contained. We'll learn more about that. Some cells do not have a nucleus. Eukaryotes do, prokaryotes do not. That's the first thing they look at. Second thing is the number of cells an organism has. If it's only one cell, it's a unicellular organism. If it has more than one, it's a multicellular. So that's the second characteristic to help break down what domain or kingdom an organism will be in. And the third one is how they obtain energy. If you remember from the last lecture, we said that all organisms, one of the characteristics of organisms is using energy. Autotrophs make their own energy. They use it, but they just are able to obtain it from the environment, and they can use it themselves. Heterotrophs, which is most animals, they have to eat in order to get that energy. And that's where that idea of producer and consumer comes from. The autotrophs, all plants, are producers. Heterotrophs, all animals, are consumers. Okay, so those 3 things determine which domain and kingdom you go into. There are 3 domains. We've got the archaea domain, which is a specific type of bacteria. We've got the bacteria domain, which is all the rest of the bacteria, and the eukarya domain, which is all the other organisms, all animals, all plants, basically all eukaryotes. Anything that has a nucleus is going to be in the eukarya domain. Okay, That's the top line. That's the broadest, least specific category. Then we get a little more specific into the kingdoms. There are 6 kingdoms. 2 of them involve bacteria. We've got the archaebacteria and the eubacteria. We'll talk about that difference eventually. It's not super important. Then we have what's known as the protists, fungus, all plants, and all animals. Okay, those are the 6 kingdoms. We are in the animal kingdom. Okay, we are animals. Some more than others, but that's still a very broad category there. I mean, do you have much in common with a squid or I don't know, an ant or an elephant, or think of all the animals that exist, and we're in the same kingdom as those. So we have to break down into more specific categories, and that's what the phylum, the class, the order, the family, the genus, and species does. And if you look at the pictures there, you can see that there's less and less animals as you go. When you get to phylum, that's known as, this is for the black bear, American black bear, Ursus americanus. They are in the phylum known as Chordata. All chordates, all animals that have a backbone, a spinal cord, are chordates. We are also in that group, okay? But you notice we've lost a lot of the invertebrates now, so we're getting more specific. You go to class, that's Mammalia, that's mammals. We would still be in that group, but we've lost anything that isn't a mammal. All the fish are out of that group now, so we're getting more specific, okay? We go to order, which is Carnivora. So you still have like dogs and tigers and cats and stuff in that group, okay? But it's getting narrower, there's less and less in there. Then we get into the family, genus, and species, and really we're just talking about bears at this point, okay? Ursidae is bears, Ursus is an even more specific type of bear, and then the species is a specific single type of bear. Now the naming, if you notice, here's the name right here. It's always 2 words, it's always written in italics, and it follows these rules of binomial nomenclature. Binomial means 2 names, nomenclature means naming. So there's always 2 names, and you notice it's the species, it's the genus and the species. This is the species right here, Americanus, and it's showing you the whole name. So the genus is the first one, and that's capitalized, and then the second name is the species, okay? For humans, our scientific name is Homo sapien. So our genus would be Homo, and our species would be sapien, okay? We'll possibly go more into that. I'm probably going to give you an assignment on this, so make sure you are up on this. I think that's it. It is. All right, you animals, I'll talk to you later.